So you guys know that when we replicate our DNA during the S phase of interphase, we get two new daughter strands, two new daughter molecules from one parent molecule. And you know that this is the semi-conservative molecule because one strand is from the parent molecule and we have one new strand. But there were a couple of different models that people came up with, and no one really knew which one was the correct one. But then there were these two researchers named Meselson and Stahl, and they came up with the correct model of DNA replication. So we're going to talk about that. So first, let's watch this video on Meselson and Stahl's experiment.
The Meselson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen.
What that means is that they were put into this petri dish, which all of the nitrogen in it had an extra neutron, because isotopes have different numbers of neutrons. So rather than having 7 neutrons, these nitrogens had 8 neutrons. So they were heavier, they weighed more. And so when these guys were making their DNA, all of their DNA had that heavy nitrogen in it because that's what the nitrogen that was available to them. And remember that DNA contains a nitrogenous base, so there's nitrogen in it. So all of the nitrogen in the DNA inside these bacteria weighed 15. The atomic mass was 15. It was heavier than normal nitrogen.
The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected.
So now that they've moved into this new petri dish, all of the nitrogen available now is normal N14. Has 7 neutrons, it's the normal nitrogen. So any new DNA molecules it's making now is going to contain the lighter nitrogen, the N14, because that's all that's available to it. It still has the old N15 inside the original molecules, but any new molecules it's making is going to have N14.
The DNA was then extracted and dissolved in a solution of cesium chloride.
Notice how the bacterial DNA was circular. Those are the plasmids that make up the DNA, the bacterial chromosome, unlike our linear chromosomes.
The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube.
That means that it just spread out by density. So a density gradient is established in the tube.
DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient.
So as we spun the cesium in the centrifuge, the denser cesium moved towards the bottom, and the lighter cesium moved towards the top. So these molecules are going to go where their density is, just like ice floats on top of water. So the lighter nitrogen that only has N4, or the lighter DNA that only has N14 in it, goes towards the top because it weighs less, and the heavier nitrogen goes towards the bottom because it weighs more, and that's where it fits in more nicely with this density down here, the heavier density. This one is less dense up at the top. So this tube only had N14 in it. This tube only had N15 in it. Those were controls. That way we could compare our experimental groups to them.
After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15.
So this is after one DNA replication. They found one band in the middle. So the density was somewhere between N14 and N15, meaning that one strand of DNA was still heavy, or like originally, and the other strand was lighter because it had been exposed to the N14.
After 2 generations in N14 medium, 2 bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So this is why After the first generation, we saw an intermediate because it has one N15 and one N14. After the second generation, it is replicated again, like it just showed you, and now we still have these two bands that have one N14 and one N15 because the original DNA strand is still in there because we don't touch it. We use it as a template, but we just keep making more and more of these N14s. So that's why we have a band up at the top that's lighter and a band in the middle, which has the hybrid, the combination of the N14 and the N15. So we can see that also here. So after one round of replication, we only see one, but then after 2 rounds, we see one at the line of the N14 and one in the middle. This is the hybrid DNA, and this is the normal DNA that's made with all N14. And after lots and lots of replications, we will continue to see those 2 parent strands with the N15. They still exist. There's still those 2 hybrids, so we still see this band, but all new DNA molecules will be made of N14 and will be at that band right there. So this is how they came up with the semi-conservative model of replication.